Hey, hello everybody. A warm welcome to this Royal Society seminar today. My name is Eric Warrant, and I'm a professor of zoology at the University of Lund in Sweden. And I'm on the academic editorial board of the Proceedings of the Royal Society B. And I'm also the chair of today's seminar. Just before we begin, a reminder to keep yourself on mute, if you could, please. Most of you are already on mute, I see, but those who are not, please do mute. This session is also being recorded, but only the speakers will appear in the recording. And please also type any questions or comments that you might have in the chat box at the bottom of your screen. If you would like at the end to actually ask Katrina questions today, you can, of course, by raising your hand in the image, the thumbnail of yourself that's going to appear on the screen. So you can actually ask with a real voice if you wish, but you're also welcome to add comments to the chat box too. As I said, thank you very much for joining us for the seminar. This is actually part of a series in which authors and editors talk about ecology and evolution based around articles published in our journals, the Proceedings of the Royal Society B, the Philosophical Transactions of the Royal Society B, Biology Letters, the Journal of the Royal Society Interface, and Royal Society Open Science. If you haven't already done so, please do register with the series at the Cassini web platform to watch previous talks and to receive alerts when a similar seminar is planned. Today, it's my really great pleasure to introduce Katrina Phillips, who's the speaker for today. Katrina is a marine ecologist with an immense passion for sea turtles, particularly concerning their movements, their migratory routes, and the connectivity that exists between different populations, which also makes her a keen sea turtle geneticist, it turns out. She's done all of her postgraduate university training in Florida, which is actually a pretty good place, I think, for studying sea turtles, which we'll hear about today. She's also earned, as she's done her training in Florida, she earned her Masters of Science at the University of Florida, and she earned her PhD at the University of Central Florida. And at that latter institution, she did the research that she'll tell us about today. That research also led her to being awarded as an outstanding graduate in biology. Congratulations to Katrina there. She currently holds a prestigious NSF postdoctoral research fellowship in biology to study loggerhead migratory genomics at the University of Massachusetts Amherst. And later this year she'll start heading backwards towards the south again to become senior marine scientist at the Dauphin Island Sea Lab in Alabama. In this seminar, Katrina will tell us about her Proceedings B article titled New Insights on Sea Turtle Behavior During the Lost Years. And if we've got time at the end, we'll answer any questions, as I said before. So you can ask those questions in real life if you wish, by raising your hand in your thumbnail. Or you can also type, as I said, your questions into the chat box during the talk, and we'll try and cover all of them, or at least as many as we can. Thanks again, everybody, for joining, and thank you again, Katrina. And I will now hand you over to her to begin her lecture. Thank you.
Thank you so much, Eric, for the introduction, and thanks everybody for being here. I'm going to speak a little bit about this paper that was part of my PhD research, as Eric mentioned, at the University of Central Florida, with my co-authors, Nathan Putman and Kate Mansfield. And so when I get started here, I thought I'd do a little bit of background to the context of what I'm talking about in terms of the sea turtle lost years. So I was going to start by talking a little bit about sea turtle ecology. And that's all through the lens of how we are observing sea turtle ecology. So just a quick reminder that this is us trying to observe what sea turtles are doing. We are mostly a land-based organism. We might have some tools at our disposal to do some research at sea or maybe from the air. But a lot of our understanding of sea turtle biology and ecology is biased by our limitation of what we can observe. So I'll kind of point that out here in the context of the lost years. So what we know the very most about is that sea turtles come up and nest on sandy beaches in many places of the world. And we're really good at counting nests and evaluating nests. And that's a very valuable part of monitoring their populations. They come up and nest. They lay about 100 eggs per clutch. Once those eggs hatch, they head out to sea, where they're in this sort of at-sea juvenile stage for a number of years. At some point, they'll transition to a larger juvenile habitat or a subadult habitat, and then transition again to an adult foraging habitat, which is at that point when they begin those reproductive migrations to nesting beaches. But in the grand scheme of the sea turtle's entire life cycle, the amount of time that they spend on land on those in that easily observable state is very small. So nesting females might spend about 10 hours on the beach every 2 to 3 years. The eggs themselves are incubating for about 2 months before they hatch. And then they head out to sea. And that's the part that I'm focusing on here in this paper. That is the 5 to 10 years we think that they're out in the stage that we call the lost years. After that time is when they transition to those other juvenile habitats for maybe 5 to 10 years. And then once they reach maturity for another 20 years or more, they're in those adult foraging habitats. So that's a little bit of the context of what I'm talking about when I talk about the lost years. And so now let's meet our cast of characters. There are 7 species of sea turtle found globally, and all of them are considered threatened and endangered at least some part of their range. For the IUCN red listing, for the global view of each species, there are 2 species that are considered critically endangered. Those are the hawksbills and the Kemp's ridleys. We have green turtles that are considered endangered. We have 3 species that are listed as vulnerable, and those are loggerheads, olive ridleys, and leatherbacks. And then we also have the flatbacks here that are considered data deficient. And so in terms of just the biology, the curiosity of knowing what they're doing during those lost years, it's also really valuable to know what is happening in those offshore stages to be able to monitor and manage these populations effectively. And the way that each species behaves in that offshore stage is a little bit different depending on the species. So a quick sort of summary of how that works. So here are our 7 species again. And then the way that they disperse after they hatch are a little different. So the flatbacks up here at the top are considered a type 1 disperser. So when they hatch, they head out to sea, and for all of those life stages, the early juvenile, late juvenile, and adult, they're pretty much in the neritic zone, which is basically over the continental shelf, which is usually defined as the edge 200 meters of depth or shallower. So they don't really head out as much into that oceanic zone beyond the continental shelf. For these other 4 species, greens, hawksbills, loggerheads, and kemps, they're considered more of a type 2 disperser. So they do have, like I showed in that diagram, that oceanic stage, and then they transition to a juvenile stage over the shelf, and then an adult stage over the shelf as well. And then for the final 2 species, the olive ridleys and the leatherbacks, they're more of a type 3 disperser. So the little juveniles will head out to sea, and then they don't really transition back to nearshore habitats after that. Even as large juveniles and adults, they stay pretty much off that shelf. So for the focus of the talk today and the paper that I'm talking about, we focused on these 4 species that are the type 2 dispersers that follow sort of what that diagram was that I showed, where they head out off the shelf for a period of time and then transition back onto the shelf as larger juveniles. And just for a little bit of context for those who aren't familiar with these species, of where are they found sort of on a global scale, the Kemp's ridleys are mainly a North Atlantic species, even more specifically, mainly a Gulf of Mexico species, but they are found along the east coast of the US and sometimes even dispersing over to the coast of Europe. Hawksbills are more broadly distributed and usually associate with reef habitats. The green turtles also associate often with reef habitats or seagrass beds. And then the loggerheads are more broadly distributed and are benthic foragers. So if you were paying really close attention to these range maps that I just showed, you might have noticed that all of the species overlap here in this North Atlantic region. And that's the region that we focused on here in the paper as well. So a little bit more about the sea turtle lost years, specifically in the North Atlantic. A lot really wasn't known for many, many years. There were some anecdotal evidence that they were associating with this floating seaweed called sargassum, which is more common in the North Atlantic. And it's a great habitat for them. They can find refuge. There's a lot of little critters that they can munch on. But how long they were associating with the sargassum, where exactly they were finding it and following it, and how their movements were, was not known at all for a really long time, or even just where they were. And so sargassum tends to form these lines and rows at frontal boundaries of different currents. And they have these little gas bubbles that keeps them at the surface. And another thing that happens to stay at the surface and kind of conglomerate around frontal boundaries is oil. So unfortunately, during the Deepwater Horizon oil spill in 2010, there was a lot of oil happening at these same areas that the sargassum was collecting and in this habitat. And as part of the sort of response to the oil spill and understanding how it was affecting habitats and organisms, many groups went out to document what was being impacted. And as part of that, they found many turtles in this lost year's life stage in these habitats near the vicinity of the oil spill. So just to give a sort of overall picture of where that was, here's a map of that northern Gulf area. It shows the area of the Deepwater Horizon platform. This sort of red shading is showing the extent of the spill overall. And then each one of these points is a turtle that was encountered and then showing its degree of how it was impacted by the oil. But many of these turtles were in that lost year's life stage that we did not know that there were so many of them right here off the tip of Louisiana. So this was a really sort of interesting data point to use going forward to figure out what is going on in these lost years. So the goal of our research was to understand these lost years turtles. And so the simplest way to probably do that would be to tag hatchlings as they're emerging from the nest and follow them from the nest out to where they are. But unfortunately, our tracking technologies, as they currently stand, aren't quite small enough to have like a remote access tracking tracker added to a small hatchling that is A, cost effective because most of them are going to be eaten, and B, small enough that it would be able to track these small guys. So what we were excited about was that knowing now that there are a lot of these lost year turtles that are relatively accessible from shore in the Gulf of Mexico, we can use that as a starting point for learning more about these lost years. So the three goals that we stated as our main goals in the publication were to characterize those lost year movements and how they relate to those traditional definitions like the type 2 dispersers, compare turtle movements to oceanographic drifters to look if there's evidence of swimming behavior or if they're just floating around out there at the whim of the currents. And then the third one is to examine possible recruitment to those nearshore habitats over the continental shelf to see more about where that transition is happening and at what size these individuals are actually transitioning from that offshore stage to the neritic juveniles. So that transition there. So the first step is to find them. So we knew from that response to the oil spill that there should be turtles out there, but it's tricky to go out into the ocean and find them. So we started our search in the years following the spill. So no oil left at this point. well, not no oil, but it wasn't a spill conditions at this point. So we started our search area around that tip of Louisiana where those individuals had been documented. We also did some trips out of sort of the panhandle of Florida and also the sort of Gulf Coast of Florida and the peninsula. And our target wasn't to just go out and find turtles, because that is probably worse than finding a needle in a haystack. These turtles are, I don't know, 20 to 30 centimeters long, and we're out driving around in the middle of the ocean. So what we did is we actually looked for those lines of sargassum first. And then once we found those, we would ride along the sargassum and then look for turtles that were interacting, swimming around the sargassum, which looked a little bit like this. So we were out on a boat, we would search for the sargassum. If we found a turtle, we would back into the sargassum, scoop it up, bring it on board, do the workup, and then repeat. And if you're interested in seeing more about what this whole process looks like, we were fortunate to have a group come out with us from National Geographic, and this is showing the areas where we found turtles. These are all the capture sites of where we managed to find turtles. And so we did have a film crew come out with us as part of this National Geographic series. And so you can see a little bit more of this, all of the searching, but they sort of fast forward through that part, and then they show what it looks like to capture the turtles too. And so there's many different stories in the behind the scenes episode. We start at about the 30-minute mark. So once we have the turtles on board, we do all sort of like standard morphometric measurements. Importantly for this paper, we also added these satellite tags, which I'll talk a little bit more about. And then we also took some genetic samples that we used as part of another study that I'll touch on at the very end. So starting with just the sort of size distribution of the turtles that we found, I'll put them up here in a moment, but to start, I just have these shaded regions, which are the estimated minimum sizes that the turtles of each of these species are when they transition to those neritic habitats and start feeding on the benthic habitat area instead of at the surface. So these are like the sizes we would expect them to be transitioning away from these habitats. And then I'll overlay that with the capture sizes that we actually had for each of the species. So you'll notice for the loggerheads and the hawksbills, we captured individuals that were smaller than what we would expect to be that transitioning size to neritic juveniles. And then for the greens and the Kemp's ridleys, we were sort of in that window of, okay, this is the sizes, the larger ones that we found at least, are the sizes that we would expect to be transitioning to those habitats sometime soon. You can also get a feel from this figure for the sample sizes that we had for each of the species. So for loggerheads, we only found 5. For green turtles, we captured a total of 94. Hawksbills were 4, and Kemp's ridleys were 28. So you might be asking yourself, why are you publishing about 5 individuals of loggerheads? But again, these are so difficult to find and know where they are that even just having information on where 5 lost years turtles were and their behavior is really valuable and interesting to present. And I'll also mention we weren't specifically targeting any of these species. So this pretty much reflects the abundance of each of these species out where we were looking for them. After we had the turtles on board and they were measured, we added these little Argos satellite tags. These are microwave telemetry, 9.5 gram solar powered tags. So the solar powered part of it means that they could be much smaller, so that these small individuals could carry them. And then we would release the turtles the same day we caught them, again into sargassum habitat. And when we released them, we would also release 2 passive oceanographic drifters, again, to look at how the turtle movements differed from those passive drifters, and if the turtles were just drifting with the currents or if they were somehow influencing their movements. To analyze the movement data, I'll talk just a little bit about what we did with the data part of it. Those Argos tags, because of the duty cycle that we used, had sort of spurts of data, which is shown here as these sort of red dots on the plot. And so we would get spurts of information, but we were interested in sort of standardizing that across the time series. So we used a Bayesian hierarchical model to interpolate the positions in between the spurts of data, sort of following that blue line, and then like confidence credibility intervals around them. So we interpolated positions at 12-hour time steps to standardize across all of the tracks. And then we used a state space switching model to estimate behavioral states at each of these positions. And so to just quickly explain what I mean by behavioral states, it sort of puts the behaviors on a scale from 1 to 2, and a sort of state 1 behavior is a quick directed movement in a sort of in a particular direction, whereas a state 2 movement is a more of a slow movement, and there's a lot more turning involved. It's not like a straight line. It's more of sort of a sinuous movement. And so movements can happen or can be categorized anywhere along that scale. And we did that for the full data set of the tracks that we had. So I'm going to put all that data up all at once to start the full data set of all the tracks and the behavior states, and then I'll break them down a little bit more. So overall, we tracked 112 individuals. The average tracking duration for each individual was a little bit over a month, 37 days. Overall, you can see that there's a lot going on, but we were surprised to see how much of the data occurred over that continental shelf, which we weren't necessarily expecting, because again, in this life stage, they're considered oceanic stage. And there were quite a few estimated states closer to that state 2, that slow sinuous movement. So we were thinking, okay, maybe this is showing that transition to the benthic habitat, benthic foraging, and they're more of like an area restricted search. And then we also had quite a few individuals that sort of became entrained in that Gulf Stream, went around the tip of Florida in this sort of narrow little, I don't know, pathway here, which is where the current travels, and then out into the North Atlantic. So we weren't necessarily expecting that either. So that was really interesting to see. So looking more at how the turtle movements and the drifter movements compared, we also did an analysis of all of the drifter movements. So again, from that state 1, that's the quick direct movements, to the state 2, which is those slower, more meandering movements. Overall, the distribution of the drifter states was sort of equal almost across all of those different movement states. And when we compared that to what the sea turtle movements looked like, we found that there were a lot more sea turtle movements in that, closer to that state 2, so those slower and turning movements. So again, we were interested in seeing what was going on there. Just to take a quick look, I didn't actually have a figure in the paper, but in the supplement, there is a figure that shows the actual drifter positions. And so the drifters moved a lot like this. So they were all the same release sites as the turtles. One thing you'll notice is that compared to the turtles, all of the drifters that left the Gulf ended up washing up on shore on the east coast of Florida and did not continue up into the North Atlantic. And this is a really interesting point too, because many of the turtles ended up sort of leaving the Gulf Stream at that point and kind of bumping up into the North Atlantic. And many of them made their way back to the Gulf Stream after that. But it's curious that at that point where many of the drifters washed ashore, the turtles kind of went the other way. So that's sort of pointing to sort of a behavioral component there. And then I'll also point out here in the Northern Gulf, how many of these drifter locations happened very close to shore, and many of them ended up washing up onshore. And as far as we know, none of the turtles washed up on shore or became that close to shore. So this is another interesting tidbit that we wanted to look more into. And so one benefit of this being a seminar instead of a paper is that I can show the movement data in movement, which is a great way to understand it. So this is an example of a deployment of one day where we had 2 turtle captures. We released them together with 2 drifters. The black lines that are going over to the left are the movements of those two drifters after release. And then the gray line and the green line to the right are a green turtle and a Kemp's ridley after we release them. And so what you'll see here is that at some point the turtles do sort of start heading in towards shore, but when they get to a certain point, they decide, no, we don't want to go that way anymore. And they turn and they leave shore. And so what we think is happening with all of that behavioral state data is a majority of those type 2 movements, that slowing and turning, isn't necessarily recruitment to benthic habitat and area restricted search, but it's a slowing and turning away from the coast to go back offshore again. So that was something really interesting. We're not exactly sure what the cues are that they would be using for that, but I'm sure it's something that'll come up in the discussion. And just to break it down by species a little bit, so each of these is showing the composite of each species, with the darker points showing those points that are over the continental shelf, the lighter points are off the continental shelf. For the green turtles, there were a lot of points. Also for the Kemp's ridleys, there were a lot of points over that shelf, again, which we weren't expecting for these oceanic stage turtles. There were lots of also points offshore, especially for the green turtles. It was interesting for the Kemp's ridleys. There were quite a few. It almost looked like they were selecting positions over the shelf rather than off. And then contrast that with the loggerheads and the hawksbills. Of course, we have a much lower sample size for each of those. But for the most part, those were spending more time off the shelf. And we also saw a few places where just given the movement data and comparing it to where drifters were coming close to shore, it looked like turtles were behaving a little bit different. That might be indicating, okay, this is a turtle transitioning more towards interacting with benthic environments. And we had one example each for the green turtles, the Kemp's ridleys, and the hawksbills of individuals that seem to be moving into those close to shore habitats in areas that drifters weren't doing that as well. So this could be showing potential early recruitment to those nearshore habitats. You'll notice I didn't mention that happening at all for the loggerheads. And again, the loggerheads stayed more off of the continental shelf overall. And that probably goes back to, if you remember, the size of the turtles that transition to those nearshore habitats. Loggerheads are generally much larger when we start seeing them there. So the turtles and the sizes that we captured are not as close to that transition stage as the other species. And so to just look at the loggerheads again, these were some of the loggerhead movements from a couple of different years kind of put together. And again, the loggerheads are staying much farther from shore and moving greater distances overall than a lot of the other species. So this was really interesting to us. We were also interested, so this is showing where they went after we observed them, but at the point that we saw them, they were already at sea for a year or two. So we were also curious, where were these individuals originally coming from? What was their rookery of origin? So we did do a separate genetic analysis to sort of analyze that, and we looked at a diagnostic fragment of mitochondrial DNA. We did that for all of the species, but we had enough of the green turtles that we could do a mixed stock analysis. So we built models using the haplotype frequencies of that mitochondrial DNA at potential source rookeries, which are the black dots on this map. We also looked at the haplotype frequencies at each of the capture locations. So for here, we focused on the Northern Gulf and the Eastern Gulf captures. And then we also included an element of the probability of being assisted in their movements by ocean currents. And with this analysis, we found that the majority of the turtles that we captured in the Northern Gulf were probably coming from rookeries in Mexico and potentially some from Costa Rica, while the ones that we captured in the Eastern Gulf were probably coming from different rookeries in Mexico, potentially also Costa Rica and as far away as Suriname. We also had that mitochondrial DNA data for the other species that we didn't use in the mixed stock analysis. And one interesting thing I'll note is that for one of the loggerheads that we also tracked, it had a haplotype that so far has only been linked to rookeries in Brazil. So again, probably a long distance transport for these loggerheads more so than the other species. So overall, we've tracked, we've learned a lot about where these turtles are, where they're moving to, at least in that 2 to 3 month time span that we were able to track them. In terms of these type 2 juveniles, one thing we mentioned in the paper is that maybe we should adjust just our perspective on what that looks like a little bit. So instead of these neatly fitting into each of these boxes, each of these life stages are probably straddling the boxes a lot more than this. And one thing we were also mentioned several times was highlighting this terminology, calling them an oceanic juvenile versus what we were proposing as a dispersal stage juvenile, which others have used as well, to get away from defining them by this oceanic zone, because it seems like even in this life stage, they're kind of heading over the shelf and off the shelf and back and forth, and they're not strictly oceanic, which maybe it's all semantics. Who really cares, right? But when we go back to the conservation and management of these species, it is important to know that this juvenile stage isn't only happening offshore. It is coming in inshore as well. And so this is a map showing the tracking data with the colors different by the species, and a couple of managed areas that are currently observed by different countries. So there's the Scorpion Reef Protected Area off of Mexico. That's where that one hawksbill seemed to be hanging out. And then there's also this critical habitat that's been designated under the US Endangered Species Act, specifically for loggerheads. And you'll notice in that sort of Gulf area, the line kind of follows the edge of the continental shelf, which actually captures the loggerhead movements pretty well. But there is a proposed critical habitat proposed now for green turtles. And the green turtles, which are shown here in green, spent a lot more time over that continental shelf in this life stage. So we did contribute data to that proposed green turtle habitat. And this is what that proposed green turtle critical habitat looks like now, coming in much closer to the coast. So we were excited to have our data incorporated into this plan. So again, in addition to just learning about the biology that we knew very little bit about before, it can be put into action to create management and conservation to protect these species. So if you're interested in learning more about anything that I mentioned today, here's a link to the paper, and I think it's linked all over the website here. So please check it out, and thanks for your attention. I'm happy to answer any questions you have.
Thank you very, very much, Katrina. That was a fascinating talk. I'm, of course, what I might do is I see if I can put everybody into some kind of gallery mode here so I can see any questions that might arise here. Yeah, no, thank you very much, as I say, for an excellent, excellent lecture. Maybe if I've got the possibility to maybe start us off just by asking for me, I must confess, I'm not an expert on sea turtle biology, but why do they make this transition? Why do they spend so much time at sea and then come back in? What is it that triggers that? I mean, it seems that some species don't even do it, some do. So what is the trigger? What is it that suddenly says, now I have to come into shore and I have to spend a whole pile of years here instead?
Yeah, well, so we think what's happening first with the transition offshore is that there's a high density of things that really want to eat little turtles very close to shore. So as they're heading to the water, there's raccoons, there's birds, there's crabs. As soon as they hit the water, there's sharks and birds still and fish. So heading out to sea, this like sort of lower density of predators. So that part of the dispersal makes sense. And then we think the transition back closer to shore for those species that we were talking about probably gets at some sort of resource limitation that they need to head. Then they reach a certain size, they need more prey for themselves. So that's when they head inshore to get to those more productive areas and then continue growing.
Mm. So how come leatherback turtles don't need to do that?
I mean, it's a great question. Leatherbacks, when they hatch, are the largest. I mean, they're the largest overall. So, and they also grow pretty quick. So maybe that might explain. Oh, so they do head out to shore, and then when they all the different species have a different sort of preferred food source, and the leatherbacks prefer jellyfish, and so the
Mm.
jellyfish can be plentiful way out
Yes.
at sea. You
Yeah.
don't necessarily have to head inshore. Yeah.
Yeah. Fantastic. Okay. Well, that's interesting. We've got a couple of questions appearing now in the chat. As I say, everybody is welcome, of course, to turn off their microphone and ask a question. It would be good if you just put your hand up first by using that little raise hand thing in Zoom. So I'll start off with the first question that's come from Leah. And I was wondering if you saw any sex-related differences between male and female dispersal behaviors.
Yeah, that's a great question. The short answer is no, because at this life stage, there are no external characteristics like sexual dimorphism between males and females. So we don't actually know which individuals were male and females that we caught. Um, we did not do like one way of looking into that would have been done to laparoscopy, which we did not do. We do have genetic samples. They don't have sex chromosomes, so we can't diagnose it that way. But hopefully, maybe one day we will get to a point that we will be able to use those genetic samples to go back and look at maybe some. I think there's somebody on the call right now. Look at maybe some differences in methylation between males and females that we can go back and then get at that question.
Right. Thank you. And a second question that's come from Emma or Emma L maybe. Do apologize if I've got that wrong. I'm interested. The question here is I'm interested in the comparison between the drifters and the two turtles in the figure. The turtles going almost the opposite direction to the drifters. Is this what you saw for most turtles? And did you expect them to be drifting with the current?
Yeah, that I mean the one that I showed was more one of the more extreme examples of the drifters doing one thing and the turtles doing something completely different. A lot of times the drifters and the turtles would kind of start heading out in the same direction and then they would sort of diverge in what they were doing. So I mean, we we did expect the turtles to move in some ways similar to the currents. I mean, even if you or I went out there and started swimming in the ocean, most of what our movement would probably be dictated by the currents. But they are showing some. There's a little bit of both. I think there's a little bit of transport in the currents, and then there's a little bit of, especially if they're heading into an area that they don't want to go to. There's a behavioral response to maybe avoid getting close to the coast or something like that. So yeah, it's a little bit of drift and it's a little bit of movement together, which I think is also really valuable. There are sort of like particle transport models to see where these individuals are going, and they're starting to incorporate this option of movement and decision making. And I think that's great. I think it's the combination of the currents and the decisions to move that will really get at what their behaviors are.
Just before I take the next couple of questions, it's interesting because from the point I work on insect migration and navigation, so and it's the same question with wind actually. How much say do migrating insects actually have in the directions that they travel in? Or are they just simply blown around like particles in the wind? And it's not the case that they're blown around like particles in the wind, actually. They do have quite a lot of say in where they go.
Yeah. Yeah.
Okay, the next question comes from Julia, who's going to ask you in real life.
Yes, hi. I'm not sure if you can see me because I cannot see myself, but I just wanted to thank you for the seminar. And I wanted to ask about Kemp's Ridley, because based on what you've shown at the beginning, it looks like the northern Gulf of Mexico, it's the only place where they actually where they actually are. So how come you guys found only, I think, four, right, or five of them? Yeah, wanted to ask about that because based on what you've shown we cannot find them anywhere else.
Yeah, no, good question. The Kemp's ridleys were the second most frequent species that we found. I can't remember the exact number, 26 or something like that.
Oh yeah, then I messed it up with loggerheads or hawksbill, but yeah, okay. Well, but still, in comparison to green turtles, there's quite some disparity. So that's would you say it's much, the 20 something that you found, or
Yeah, I mean, we were excited to find those. And again, going back to that sort of the conservation status of each of the species, the green turtles are kind of at that global scale, they're considered endangered, but actually in the North Atlantic, we're experiencing kind of an increase in green turtle nesting. So I think there's a lot of green turtles getting contributed to the juvenile stock right now. Whereas the Kemp's ridleys are not quite so much. They're the ones considered critically endangered. So there's probably fewer individuals getting produced for us to even observe. And then also going back to where these individuals are coming from. So the Kemp's ridleys basically only nest in Mexico, and then a little bit of nesting into Texas. So there's only a couple of points where they would be coming from, whereas the green turtles have more sites where they are nesting. So it's more possibilities for where they might be coming from.
okay,
Yeah. Thanks for the question.
Thanks, Julia. Okay, the next question is actually from Leah, and then the next after that is quarantine. So Leah.
Hi, thank you so much, Katrina, for such a great talk. And thank you for your answer to my question about the differences in males and females as well. I know that the olive ridleys weren't included in your study, but just listening to your explanation about the dispersal behavior of leatherbacks, I was just wondering if you could maybe comment on why you think the olive ridleys would be having this different dispersal methods as well.
Yeah, I don't know. That's a great question because they do have more of a benthic foraging when they reach adulthood. So I don't know why. I don't know why the olives would be more of that type 3.
Okay.
I mean, I guess they do. I'm trying to put together an answer for you. They do have more of a global distribution, even though they don't happen to occur like in the North Atlantic where we were. So maybe it has something to do with that. I don't know. That's a great question.
Yeah, it's interesting because I'm studying olive ridleys at the moment, and we're looking at exposure to plastics, and I'm just wondering if maybe that would have like an impact on their kind of level of spatial encountering of plastic items as well, if they're not really coming back to the coast as much. But yeah, don't worry, I might send you an email at some point.
Yeah, please do. Yeah, and
Right.
I don't have any data on them in the lost years, sadly, but hopefully we'll get there at some point, and then we'll know more about how long they're even spending in each of these habitats, which would help with when they're encountering the plastics too. Yeah.
Thank you so much.
Yeah, great. Thanks for the question.
Thank you, Leah. Okay, and the next question actually comes in the chat, pardon me, from quarantine. How far from the coast did you catch young juveniles in the sargassum mat? And considering that current collection efforts, as explored in the French Antilles and Mexico, only a few miles from the shore, could raise concerns about potential impacts on sea turtles if expanded further offshore.
Yeah, so to get out to where that sargassum habitat was, we did head out I think the closest observation we had was 20 to 25 kilometers, and we were out like 75. Now I'm switching to miles, sorry, but like 75 to 100 miles offshore. So we were pretty far out from shore. I'm not sure what you mean by the current collection efforts. I know in some areas, I mean, the transition from like the continental shelf isn't as wide, so you can get to that deep water habitat much closer to shore in some areas. And then the potential impacts. So, I mean, yeah, that's one reason why we're interested in having that sort of critical habitat, at least defined in US waters. That would dictate that if there are impacts by federal activities, they'd need to be mitigated. So like for oil and gas exploration, if there are impacts happening to individuals in those areas, they would need to be mitigated. So I could see like oil and gas exploration, or even offshore wind could come in contact with these turtles. So it would be helpful to know where those areas of interaction would be when moving forward with those activities. I hope that answered your question.
Thanks, Katrina. Thank you for the question. Our next question is actually a live one from Thomas Dellinger. Please, Thomas, go ahead.
Okay, can you hear me now?
Yes.
Okay. Nice talk, Katrina. I just posted here, if you're interested in collaborating with the other side of the Atlantic, please feel free to contact me via email. We sit at Madeira, which is about the center of the juvenile pelagic or oceanic stage. And right now we are also interested in how on these movements, both the very small and the very large turtles. But that's why I was asking, what's the maximum size of your 5 or 4 loggerheads you had?
Oh, I can't remember off the top of my head. None of the turtles that we had were over 30 centimeters SCL.
Yeah, 30 is quite big right now. So we are way over. We have much smaller here than that. So since I didn't see any track really going offshore, so you're capturing only part of the behavior really in your paper, not the whole thing. Right now, we have a second phenomenon, which might be interesting. Did you catch all the loggerheads in Sargassum or not?
Yes, we did, which doesn't necessarily mean that they're only in Sargassum, but that is where we found them.
No, they're clearly not always in Sargassum. At least I don't know about the very small ones, but the bigger ones might be. The bigger ones certainly are not. But right now we have a phenomenon this year and last year, for the first time, that Sargassum mats actually reach the Eastern Atlantic. So they're coming to Madeira and the Azores. So it's very interesting right now to really see how that affects the behavior and the transition. We will probably be putting a satellite transmitter of a large turtle to see the back migration in this year. But so yeah, it's very interesting to see these behaviors and these differences.
Yeah, no, yeah, great. And I'd be happy to connect. I'm curious, do you ever see any Kemps over there?
We had one Kemp which died on us. We had the first olive ridley. We haven't published it yet, which is the first sighting for Madeira. But we had a few hawksbills and the other species. Kemp ridleys is actually, Madeira is recorded for Kemp ridleys historically. So there are a few 100-year-old turtles conserved in the museum here. But we had one turtle which actually went to Porto, so continental Portugal, and we recovered it here, tried to recover it, but it died then of pneumonia. So there was bad luck. It was was a unique chance, but no way. So the probability, given the population size and the size of the ocean, is very, very low to actually get hold of one. I do believe that they will come here eventually if the population grows larger, that we probably start seeing them here.
Yeah.
But it's very interesting. The question is, which juveniles actually go oceanic and which don't, and why. And so there are lots of really unanswered questions. And I liked very much your approach of the drifters. I thought of it, but lack of funding did not. It was more important to know what the turtles were doing than what drifters would be doing.
Yeah, of course. Yeah. And that's where some of that particle modeling can come in too. But one other note on something that you said, I would suspect that some of the turtles and especially the ones that we had leaving the Gulf probably have connectivity to your area, but we were a little limited in the tracking duration
Right.
because
I
we
saw
didn't-
2 months.
Yeah, we didn't want these to be any sort of permanent tag attachment. And these turtles
Right.
are growing so fast that if they stay on too long, their carapace actually starts to grow around the tag. So we used sort of flexible epoxy that was meant to shed within a few months. That's one of the co-authors, Kate Mansfield, did a lot of testing on that. Because
But
we
it
didn't
usually
want them
does
to stay
because
on.
they grow and then the epoxy gets loose. We did
Yeah.
quite a bit of tagging and TDR
Yeah.
tagging and stuff here, so that was different. But I suppose that your juvenile turtles will have to go almost up to the Great Banks before they cross over to the other side. So I think the main route for the juveniles is over via Great Banks, Canada, and then start crossing. So I think Great Banks is one of the critical habitats to check for changes or alterations of turtle behavior.
Okay. Yeah. I'll definitely reach out. Thanks for your questions.
You're welcome. Bye.
Thank you, Thomas. We also have a comment actually that has arisen as a result of Leah's question, or one of her questions, regarding whether or not competition might come into play with the different locations for the olive ridley turtles.
Yeah, no, that's a great point. I would trust anything Genius says, so.
Yeah, so I guess the answer is yes.
I know much less about the olives. I don't have them in my study area really. So yeah, I trust you more on those.
He put up a heart, so
Yeah.
okay. I don't see any other hands up. Well, actually, I see Thomas's hand is still up, but I suspect that's probably because it's not taken down yet. But or did you have another question, Thomas?
No, sorry, I don't. I just don't know where to find the button to lower the hand.
No problem.
It's a common problem. I don't know where half these buttons are either. Wonderful. Okay. Any other questions anybody's got to ask? I don't see any hands up and no more comments coming in. One of the things that interested me, if I've got the arrogance to be able to ask a question as the chair, I'm very interested in animal navigation, and I saw that a lot of your turtles were navigating. You mentioned this also in your paper. And it seemed that most of that navigation was most obvious, at least, beyond the shelf out in the open ocean. Is that a fair comment, or do you see the navigation even across the continental shelf when they're in shallower water?
I mean, I think there's a little bit of both going on. And there was another paper out that was kind of even commenting on it. Looks like perhaps the Kemps were navigating more to stay within the Gulf than perhaps the Greens were. So, I mean, we do know that sea turtles have the ability to perceive and navigate using the magnetic field. So there's probably a component of that going on for sure. And then I would also suspect with those individuals that were turning away from the coast that there's something else going on as well. And I don't know if it's a response to depth cues or an olfactory cue of getting close to shore. But I imagine it's sort of a amalgamation of different cues that are all coming together to help them figure out where they want to be and where they don't want to be.
Yeah, well, certainly navigation is a multimodal thing. So,
Yeah.
And they do have a very well developed magnetic sense, but they do have a very good olfactory sense too, and not bad vision. So, yeah, there's probably a lot of factors working on this topic. Okay, do we have any final questions from anybody? Looks No hands up and no comments in the chat. And we are nearly out of time actually anyway. So maybe we should maybe call it a day. And thank you very much again, Katrina, for a very nice and quite remarkable talk on a very, very lovely paper that I was fortunate enough to be chosen to be the editor of for Proceedings B. It was one of those papers that was easy to handle because not only me but also the reviewers were very positive, and it's understandable today why. Thank you
Yes,
very, very
well,
much,
and the
Katrina.
authors are very thankful for that.
Yeah.
But thank you all. I'm so glad that we had the opportunity to chat today, and thank you all for coming and asking some great questions.
Yeah. Thank you so much, Katrina.
Thanks.
Yeah. And thank you everybody for attending.